Once we have generated phylogenetic trees from our given data, last step is to find out evaluation of our generated phylogenetic trees. What does this evaluation means? Evaluation can be defined at two levels. Number one, if those generated trees are accurate. Second one is if those generated trees are efficient. Efficient means with respect to time and memory constraints. There are four different methods for tree evaluations, which are randomized tree, randomized character data, bootstrapping, and likelihood test. In first two, randomized tree, we generate different trees which are of course random from given data, and then in the second type we have different characters which are randomized from the provided data. Third one is bootstrapping, and finally we have likelihood test, in which we check out that what is the maximum likelihood of a given tree from the provided data. What is bootstrapping? इस मॉड्यूल में हम bootstrapping को जो एक्सटेंडेड मैकेनिज्म है फॉर इवेल्युएशन ऑफ ट्रीज फोकस करेंगे और देखेंगे वो कैसे परफॉर्म की जाती है. Moving on, number one, what is the criteria for evaluation? How do we evaluate a phylogenetic tree? Number one thing, do we want to measure the accuracy of the tree? Is this the question? How to measure accuracy of the generated tree? Number thing is, the answer is that it should be consistent, it should be efficient, and it should be robustness. Robustness means that it should capture complete information from our provided data. And then accuracy of the tree building approach is important, or accuracy of a particular tree is important. That is a question which we have to answer कि हमने जो अपना ट्री जनरेट किया है वो इम्पोर्टेंट है या जो हमने अप्रोच है वो इम्पोर्टेंट है. लेट्स स्टार्ट फ्रॉम बूटस्ट्रैपिंग देन. So what is bootstrapping analysis? Number one thing, basically it is not a technique to assess only the accuracy of the technique or the tree. Roughly idea इसका क्या है? What we do, we have some given data, we have generated phylogenetic tree against that data. Now what we do, we take some artificial data and we try to build trees from that artificial data. अब हमारे पास दो टाइप के ट्री हैं, एक वो जो रियल डाटा पे हैं, सेकेंड वन जो आर्टिफिशियल डाटा के हैं. और फाइनली हम क्या करते हैं, वी ट्राई टू कंपेयर दीज टू ट्रीज. सो if those trees based on real data and artificial data they are equal or similar, then we say that our generated tree is very good, otherwise it's not good. So what are the steps then? For artificial data, what we need a model which can generate many number of trees. So that is one step, and once we have many number of trees on artificial domain and similarly on real domain, then we try to compare them. So what is happening here is, for bootstrapping, if I said for definition, I would say that it allows an inference of the variability in an unknown distribution from which data was drawn. So what happened? We have data and we have unknown distributions, like we have many trees available from that artificial data. Moving on, if I say procedure, what will be the procedure? Again, procedure will be simple. Number one, we will try multiple sequence alignment, and then we will see out which are the common sequences. Then we will make our tree, which is again a common thing, that first we have done multiple sequence alignment, we have generated the tree, then we will generate some artificial data which would be of the same size as the original data, and then we will do MSA and tree building from that artificial data. And this replacement operation is there. So replacement operation means that we have individual columns which can apply or which can appear on multiple times, or they can disappear altogether. So concept is, a tree is generated from randomized or random data set, then large number of bootstrap replicates are generated, so which is artificial, then bootstrap trees are compared against the real data. If this information is high, this frequency, this matching information is high, that we say that this is a very good generation of our phylogenetic tree. So for a demonstration purpose, I am showing an example from Megaforce software. So what it will show, that percentage of times that a given clade is supported inside a phylogenetic tree, if that percentage is high, for example in our example, if it is more than 70%, we will say that it is a very good estimate. If it is less than 70, we say that it is not a good estimate. I will continue with this example. What is happening here? So for this thing, if I have these three alpha globins for horse, dog and kangaroo, I will say that this information is 100%. So this generated result is 100 for all these three alpha globins. But if I only see for alpha globin for horse and dog, it would comes out 69. So this 100% result is better than 69 based on bootstrapping results.